Hello everyone and welcome to today's webinar, Improved Quantitative Understanding of Oncological Replication Kinetics Using Molecular Combing. I'm Mark Lynch of Genomic Vision and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Genomic Vision. To learn more, visit genomicvision.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. And you may submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Mario Avarello, Product Manager at Genomic Vision. Mario, you may now begin your presentation.
Hi, Mark. Thank you so much for the kind introduction. Welcome, everybody, to the third seminar today. And I'm going to discuss about the improved quantitative understanding of oncological replication kinetics using our molecular combing technology. This is the third part of our summer webinar program. Where actually in the first part you learn about molecular combing in general and how are the applications that can be used with it. So in the second part we actually learn how to use molecular combing with our unique genomic Morse codes to make a molecular marker to see large genomic rearrangements. And today we're actually going to go through the use of molecular combing to understand the replication kinetics and the mechanism of replication kinetics in cancer biology. So the difference of it in normal cells and in cancer cells and how this can be used with protein modification or drug screening. And in the next seminar on July 1st, you actually will learn what is the use of molecular combing in gene editing to qualify and quantify the genomic rearrangements happening after gene editing, after the use of CRISPR-Cas9, for example, or doing biomanufacturing for therapies. So today, as mentioned, we will focus mostly on oncology and we will focus to understand how the replication kinetics are in cancer cells, how to compare replication kinetics between normal and cancer cells by using our unique replication combing assay. So this is indeed used to see accurate measurements of DNA replication kinetics, to see the replication effect of rearrangements in target genes. And this will actually bring to the drug development by the identification of the replication stress events and drug screening and efficiency at the very end. So indeed, the agenda of today is actually divided in two parts. The first part is going to be based on the principle of our replication combing assay in our molecular combing technology. And the second part is to actually walk you through what is the use of our RCA and single DNA molecule technology for drug discoveries, screening, and then at the end for treatment refining. So, but now let's start immediately and go to the principle that are leading the RCA in single DNA molecule resolution. So as everybody knows, there are different phases in the circle of life of cells. They're going from the G1 to the duplication of the genome that then goes to mitosis, so for dividing these into the two daughter cells. With molecular combing, we actually will focus on the DNA replication itself. So basically, we will see what are the replication events that are occurring during the S phase of a cell. And this is actually how our replication combing assay look like. In this cover slip, you can see here in blue the single-strand DNA fibers, and in red and green the replication tracks. I highlighted with the white arrows the origins of replication as well. So basically, you can already see what is the power of the technology, because we can actually detect the bubble, so the bidirectional fork, already in a single DNA molecule resolution. And this is a very powerful resolution for our replication combing assay. But now, how to work with it? What are the different steps that are walking you through the use of the replication combing assay in combination with molecular combing? So in the step one, there is the pulse labeling. So the pulse labeling is indeed to pulse cells with two different thymidine analogs that are here CLDU and IDU. So these are modified analogs, the NTPs, in order to have a chlorum or a yodium on top that can be used for the visualization later on. So basically, we have in this schema here three origins of replication, the three gray circle, and we know that especially in human cells, the replication is asynchronous, so not all replication are fired at the same time. So basically, we will pulse for 30 minutes with the first nucleotide. In this case, it's CLDU in red. So what we will see is actually that the replication has been fired already on the first bubble in the time we use the red. So while it's exactly at the same time when we use the red in the second bubble, and the third bubble hasn't been fired yet. So then we wash out the CLDU and we use the second pulse, that's IDU in this case, for more 30 minutes, and we'll see in green. So then we can see the continuous of the tracks from the replication forks in the first bubble, in the second one, and then in this case, we'll see also the third bubble that will be fully green. So in this case, we go to what is actually the replication tracks in a combing. Indeed, we use a counter staining to visualize the single-strand DNA in blue, and then in red and green, the replication tracks. And this brings to identification of the timing of which an origin has been fired at that specific time of 1 hour, 60 minutes for the two different pulses. So then by using our replication combing assay, we can actually then take measurements of each single track in order to have a precise statistical analysis. So the replication combing assay, or assay, is a powerful technology that will actually bring more detail than the normal used technology in the lab. So indeed, RCA allows to have 7 different kinds of signals according the actual bubble firing, so in the timing that previously I showed. Indeed, we will have the initiation 1, so where we have the blue in the middle, then the 2 red tracks, the 2 green tracks, and the 2 blue extremities. So this is actually a replicon where we can clearly distinguish one replication forks on the right and the other replication forks on the left. Then the initiation 2, well, we actually see that there are the the green tracks and the red tracks and the blue at the end. In this case, the replicon is the second origin that I previously shown. So it started exactly at the same time when we use the CLDU, the red one. So for this case, you don't see the blue in the middle of the red, but we can still see the two forks on the red and on the right, on the left and on the right. And then later on, we can also see the initiation 3. So this is the latest that has been fired because it contains only the green tracks, so only the second pass and not the first one. So these are three different kind of initiations, but with combing, we can also see termination. Indeed, termination is shown as a flipped color compared to the initiations because we consider that the bubble is going to fuse together when the replication goes at the end. For this case, we have the termination 1, where we actually see only red tracks. And in this case, we consider that the green track has been fused together, so the replication has been resolved for the first for the second pulse. So now we only see the first one, that is like the red. Then we have the termination 2. In this case, we have the green track in the middle and the red at the extremities and then the blue at the extremities. So these actually bring us to see that the replication, the green are actually going to fuse together and after the red is coming. And then we have the latest that has been fired again in time of replication firing. So, well, it's called termination 3, where we still see a blue in between. So the green are going to, this would take some time to fuse together. And finally, we can see the unidirectional fork. The unidirectional fork are basically those forks that went so apart each other from the other fork on the other side that is considered at this time unidirectional. So the other part, the other strong part of our RCA is actually that we can see always the blue or the single-strand DNA at the extremities of the replicons. And this actually brings us to avoid any bias of DNA breakage into the analysis by using our molecular combing technology. So by RCA, we have 7 different kinds of signals. We avoid to have a bias coming from the DNA breaks that might be introduced by the stretching forces that are applied to the DNA itself, because we only take for statistical analysis the intact signals. So in this case, we can actually use the parameters, different parameters, to understand the mechanistic role of the replication kinetics. So we can basically see at the fork speed. So in this case, we have a fork speed of unidirectional or initiation 1 in the picture below. What we actually do is understanding or calculating the speed of the 2 tracks, so the green and the red, or the green and red for both forks, so on the left and on the right. And then we can see if there is a shorter replication going on a specific DNA fiber, like the one that is shown in the picture below, where we can see that the 2 green are actually smaller than the 2 reds in this case. Then we can also see fork symmetry, so basically whether the symmetry of the right fork is the same of the left fork or vice versa. So in this case, we can see 2 different pictures again. In one, you can clearly see that the fork on the right is definitely smaller. It's asymmetrical with respect to the fork on the left. While in the second picture, we actually have 2 replicons. So we have 4 different forks. And you can see that the replicon on the left, there are the 2 green tracks. So two forks that actually are proceeding slower because the fork speed of the green track is actually smaller, but the replicon is still symmetrical. But if you look on the right side, the other two forks, you can see that the fork on the right is actually going slower because the green is smaller than the red. But on the fork on the left side, the green is not there anymore. So in that case, we consider that this is a fork stalling. So the fork has stalled exactly at that point when we use the second pulse that is actually the green. Then we can also see replicating DNA. So we can take the amount of DNA fibers or single-strand DNA fibers that are on the cover slip, and we can also calculate the amount in number kb of the replicating DNA that are all over the cover slip. And with this, we can give a statistical analysis of what is the replicating DNA for a cover slip. Just to give you an idea, on average, for a replication combing assay, we use 10,000 cells at the beginning in a plug, and from these 10,000 cells, each cover slip will have something about 3,000 signals from forks. So this gives us a large number to play and work with in order to have the replicating DNA. And finally, we can also give the inter-origin or inter-termination distance. So these are distance, for example, between two different initiations, so between two different origins, or between two different terminations. And they're very useful to understand, for example, the efficacy of a drug, because if we consider that we go in a fork slowdown or even stalling, what the cells does normally is actually fire a dormant origin. So in that case, the inter-origin distance becomes shorter. So you can already see from these few parameters and this few picture that I gave you what is the strength of our molecular combing technology used with the RCA, with the replication combing assay. Indeed, we can go with long fibers, so we have up to 3 megabases. If we compare, for example, with the used technology that is called fiber spreading and has been used in the lab since the latest 70s, but the comparison between the two technologies brings a clearly advance to the use of genomic vision technology itself, so molecular combing together with the RCA. So we have indeed a cost extraction factor, and this brings us to have parallel fibers that can be visualized in single-strand DNA and with the fork tracks on it. So this will bring to complexity of signals, having 7 different classification. RCA will then allow to have many parameters as fork speed, fork symmetry, number of origins, replicating DNA, inter-origins, and termination distance. We can distinguish between intact and not intact signals in order to avoid any bias coming from the molecular combing itself. And again, we have also for the replication combing assay, a specific module for our Fiber Studio software in order to have standardized analysis. And to perform the RCA, our equipment and our consumables are very crucial to be successful in the performance of the replication combing assay. Indeed, you want to use our fiber prep kit for the DNA extraction. It's a peculiar extraction kit that we use plug, so the cells will be embedded into the plug, and in order to preserve the integrity of the DNA molecule itself. Then the DNA will be combed on our cover slips that are chemically treated in order to allow to bind the surface only at the end of the DNA itself. And then our fiber comb machine will actually apply the force to stretch physically the DNA molecule on the cover slip in order to have parallel long fibers in a native form. And then we use our Fiber Vision S. It's an automated scanner that will take 25 by 25 picture and stitch it together in order to digitalize the physical cover slip in a digital version where all the signals are detected for the replication itself. And finally, as mentioned before, we have our Fiber Studio analysis software to actually detect and help you into performing the analysis for our replication combing assay. So we are at the end of this first part of the seminar. And we spoke about the molecular combing. There's a unique tool to understand the replication kinetics. We actually saw what is the efficiency of replication that can be understood by using our RCA and what are all the different parameters that will give a deep understand of the mechanistic events that are happening on the replication itself. So that will bring an understanding of the replication kinetics and a comparison between different kinds of cells, for example, normal and cancer cells, and then the parameters that are detected and analyzed by our Fiber Studio software in order to help in your analysis and make it more standard. By this, we move to the second chapter, to the second part of this seminar, where we actually will focus more on the applications of our RCA into the drug discoveries, drug screening, and at the end, this can be used for refining of a treatment. As everybody knows, since years, the different kind of biomarkers or proteins has been used and has been targeted in order to develop drugs that are anti-cancer drugs. These actually fall in many pathways of the cell itself for the DNA replication and repair system. So that can be the homologous recombination, the non-homologous enjoining, and some other pathways. For example, the most known nowadays is PARP1, but there are many others as Pol Theta or BRCA1 gene that actually has been used. So all these biomarkers involve the use of replication machinery, and thus their effect can be efficiently quantified by the use of replication combing assay. Indeed, if we work at the principle of RCA in drug discoveries, we have neoplastic cells that have a supernumerary of origins of replication that brings to have more replicons and replication fork. So depending what is the kind of treatment against what kind of biomarker, what is happening that you can impede the origin of replication itself, that will bring to less replicons, or you would like to work on the fork itself. So all of these can be seen and visualized and analyzed by our replication combing assay, and our Fiber Studio software will actually help you to standardize all these analyses in a way that the software itself can detect all the replication forks and give already whether these are initiation 1, 2, and 3, or termination 1, 2, and 3, or unidirectional, and the user has to approve or discard the signals, and at the end, a report will be generated, as you can see in the point 3, with all the quantification of the drug efficacy for example, the fork speed on the interorigin distance, when is in combination with a protein mutation and a drug using. In this case, you can see, for example, the blue versus the red one, and also for the interorigin distance. So by this, you can actually screen and quantify what is the efficiency of a specific treatment or a protein modification or combined together. But then how to look and how to design an experiment in order to understand the efficiency of a drug, or especially when it's also combined to a protein modification. Of course, knowing what is the target of the drug, you can design different kind of experiments. For example, you want to see at the replication for kinetics, so use the drug for the entire experiment, even within the two pulsing, or you use only with the second pulse. In this case, you will bring to shortening on the second track, so on the green one, and you will see basically that the fork speed is actually changing between treated and untreated cells. Or you might focus on restart studies, so you want to see whether the fork will restart upon some treatment or will still stall. So you can use the drug with the first pulse or in between the two pulses, as used, for example, classically with afidicolene or hydroxyurea. So what you will see is like restart of replication. So in this case, the green is actually smaller than the red, and this is because the effect of the drug is actually of our restarting after the washout of the drug. Or the drug will actually bring to the stalling of fork, and then you will just visualize the red track alone, or starting of new origins. So the firing of new origins that will be in green. And then as third, you will like to see, for example, the resection studies, whether the drug is bringing to the reverse fork. In this case, the drug is going to be used at the ends. So, and then using the exopolymerase activity of the replication machinery, the green, the IDU, the NTP basically will be taken off from the replication. So the signal will get shorter for the IDU, for the green. And these you can see in the picture here beside, where the two green are actually shorter than the red signal itself. So this is applicable for many biomarker and many drugs. For example, the PARP inhibitor, to see whether there is resistance onsets to PARP inhibitor. The resection study might be analyzed, or can be check one to see the firing on new origins, and so on so far for all the other biomarker and targets that are actually built as new anti-cancer development drug. Just to give you a few examples of the possibilities, in this case we have in the first upper panel experiment of where fork speed has been taken after the cells has been treated with niraparib or cisplatin in a combination of a protein modification that the FAM 111 in order to bring to a default HR pathway. So in this case, you can see the default itself, so the protein mutation itself, they actually will not bring to changes in the fork speed. But then when on top niraparib is used, you can see already the effect of niraparib just on the fork speed itself. And then when cisplatin is used, you can see what is the effect of cisplatin itself. While in the lower panel, you actually see a study of the resection, and in this case, it is a new compound against P53, and you see again the fork speed, how it changes. And then this has been done using the drug at the ends on the left panel, or at the beginning on the right panel. And then you can also find the quantification, whether it's used at the beginning or at the ends, with a strong effect on the fork speed only when the drug is used actually at the ends, so of the second pulses. So this brings basically to resection studies of the fork itself. So all of these can be achieved by using our equipment and our consumables. So just starting from few cells, so 10,000 cells are enough to have replication fork statistics or using 5 ml blood. So then the DNA is stretched by our fiber comp on our engraved cover slip, and the digitalization of the cover slip and visualization of the signals is going to be done by our Fiber Vision S or Fiber Vision machine. So these are two automated scanners to digitalize the cover slip. And at the end, our Fiber Studio software will actually standardize and help you to make all the analysis for the replication kinetics in all these complex parameters that can be used in replication kinetics studies. By this, we are at the end of the second part of this webinar, so where we actually discuss about the parameters that can be analyzed by our Fiber Studio software. What are the mechanistic aspects of DNA replication can be quantified once cells are under certain circumstances, basically once certain kind of treatment is done on a cell. So how RCA can be combined with different kind of compounds together, or with the different kind of compounds and mutation together, in order to understand and quantify the efficiency of the drug or the mutation itself, and then using the RCA as an external biomarker for understanding and quantify the possible drug efficiency, and then refine, for example, the treatment itself. By this, we are at the very end of this webinar. I would like you to remind that this was the third part of a 4 program webinar, where in the next session on July 1st, the quality control advancements in therapeutic genome editing and biometric manufacturing is going to be shown by my colleague Iman Mesteri. By this, I would like to thank you all for your attention, and I will be happy to take your questions. Thank you so much.
Thank you, Mario, for your informative presentation. We'll now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. With that, let's get started. Our first question is related to statistical significance, and the question is, how much statistical significance is required in the RCA application?
Hi, Mark, and everybody. So thank you for the kind question. So as we showed before in our presentation, RCA, it's really robust methods. So indeed, starting from 10,000 cells, we have something about, on average, 3,000 signals from folks on a single cover slip. And we actually did the analysis, and we saw that the 300 signals from folk speeds are more than enough to actually have statistical significance. Basically, with 300 signals reviewed, the statistical, the STD is actually not moving anymore. The statistical deviation is actually pretty fixed.
Okay. Thanks, Mario. The next question is, how can replication be used to understand DNA damage?
Well, thank you. This is a very crucial question. Indeed, DNA replication is impacted by the DNA damage, as we all know, that any DNA damage can bring to have falling fork or introducing some slowdown in the fork itself, that then can be actually visualized as an effect of the replication kinetics, as shown, putting together the different parameters that we have. So the fork speed to understand whether the fork is stalling or is actually slowing down, the inter-region distance or intertermination to see whether dormant origins are fired in between two forks. Then, this is a clear sign of the DNA damage going on and the effect of the DNA damage on the replication itself.
Okay, thanks, Mario. The next question that's came in is, can this application, and I think that's of molecular combing, be used for circular mitochondrial DNA?
Well, in theory, yes, this can be used. Indeed, we actually have DNA molecules that are in the native form, and replication tracks actually show on it. So what can be used is actually a series of probes to identify sequence of interest, for example, any locus or any sequence of the DNA where the replication want to be understood. Or we can also, by using probes, indeed identify other organelles like mitochondrial DNA and the replication going on in the mitochondria, or using the GMC, as we saw in the previous webinar, we can also see what are the rearrangements, for example, in mitochondrial DNA itself.
Yeah, perfect. Thanks, Mario. So, Mario, we're not showing any other questions at this time. Do you have any final comments for the audience, Mario?
I would like to thank you all for the attention to the presentation and the kind questions. I would like to remind you that the replication combing assay is a very robust and precise method that can be used for the drug screening, and then in a certain instance also to refine the treatment for patients. And with this, I just give you just my best wishes to everybody, and thank you again.
Thanks, Mario, for your time today and the work that you presented. We'd also like to thank LabRoots for facilitating today's educational webcast. Before we go, I'd also like to thank the audience for joining us today and for their interesting questions. If there are any other questions, they can be submitted during the on-demand period also, and they'll be addressed via Mario and the contact information that you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. We look forward to seeing you at part 4 of our webinar series on July 1st, which is really related to the quality combing assays offered by Genomic Vision that are important in understanding cell line authentication during gene editing studies, as well as bioproduction for the production of biologics. Thank you so much and have a good day.